An introduction to bioinformatics. So, aims and objectives for this session are to introduce some basic concepts of bioinformatics. First of all, what is bioinformatics? To look at homology and its importance. To look at data storage, how we manage bioinformatics data. To introduce some concepts of sequence alignment and data searching in order to identify homologs. And finally, just to look at annotation, the human factor in all this. The objectives for the session then are that you'll be able to define what homologs are and also orthologs and paralogs, give examples of databanks and show the difference between a database and a databank and between a databank and a search tool. Be able to describe the basic idea of sequence alignment and the differences between global and local alignment, describe the principles of identifying homologs and functional motifs, and the statistics that are relevant to this, and finally discuss problems associated with annotation. So the lecture is going to be divided into two parts, and in this first part we're going to look at what is bioinformatics, the importance of homology, and data storage. What is bioinformatics? Well, everyone's definition is different, but the simple definition is the application of computers to problems in biology. And we can split that up into aiding the biologist in creating, storing, searching and analysing biological data. And particularly we're referring to sequence data and to structural data, but also these days to the huge amounts of omics data that are available, things like genomics, transcriptomics, proteomics and so on. Presenting those data in a way that biologists can then use, and really the web and bioinformatics have grown up as technologies together, so an awful lot of bioinformatics tools are accessible over the web. And finally, applying the analysis that we've done in order to be able to make predictions. So just a bit of background regarding the amount of data that we have. Traditionally, biology was really quite a data-poor subject. People would go out and draw pictures of furry animals or plants, and it was very descriptive. But now biology is overwhelmed with data, and really probably only second to things like astronomy and nuclear physics in terms of the amount of data that we have. If we look at the human genome, that's about 3.2 gigabase pairs or 3.2 billion base pairs in size of DNA, and that's enough raw data to fill around about 5 CD-ROMs or 1 DVD. The DNA sequence databanks are now 9,914 billion base pairs in size. So we passed a threshold of 100 billion base pairs in August 2007. Originally, the databanks had said we passed that 2 years previously, but they changed the way they did the counting because there were some duplicates in that. So 2007 is taken as that kind of threshold. So our current count compares with 6,300 gigabase pairs or billion base pairs in January of last year, and that's twice or almost twice the figure that it was a year earlier. So you can see how rapidly it's increasing. So you can think of this as being like having sequenced the human genome just over 3,000 times. So a huge is a human genome equivalent. Now that may seem strange to you, because you may have heard of projects like the 1,000 Genomes project, and then there's the UK 10K project to sequence 10,000 genomes, and currently ongoing the UK 100K project to sequence 100,000 genomes. Well, of course, with sequencing the human genome, we don't need to store the same sequence multiple times over, because most of the human genome will be the same between individuals. So all we do is use a reference genome and then store the differences when we do this mass sequencing of thousands and thousands of individuals. We can also think of the amount of data that we now have as being like 3,200 years worth of issues of the New York Times. And the New York Times obviously is a large newspaper, particularly at weekends. That's just DNA sequence data. In addition, we currently have structures for about 151,600 proteins, and that was only 147,500 a year ago. So you can see this is still going up rapidly, but nowhere near as rapidly as the rate at which the sequence data are accumulating. So DNA data on average doubles every 18 to 24 months, whereas structure data doubles approximately every 6 years. And these are somewhat old graphs, but they show you the sorts of trends that we're talking about here. Now, this graph on the top left shows you in the mauve GenBank, yellow is EMBL, and the burgundy colour is DDBJ. So these are three resources where DNA sequence data are stored. GenBank is the American one, EMBL, now called EMBL-ENA, is the European one, and DDBJ is the Japanese one. Now, in fact, these three resources exchange data with each other on a very regular basis, so they end up containing essentially the same information, but they use different formats, they have different tools to deposit data, and different tools to analyze the data. Now, the Protein Databank, which is where we store primarily protein structure data, but also things like DNA structure and some other things as well, contains nearly 153,000 X-ray structures, about 13,000 solved by NMR, and nearly 7,000 solved by electron microscopy. But this only represents somewhere between 63 to 64,000 sequences if we're looking at less than 95% sequence identity between sequences. And consequently, the percentage of sequences for which structures are available is very small. Coming back to the human genome then, as I said, this is about 3.2 gigabase pairs, or 3.2 billion base pairs in size. That's about 30 times the size of something like C. elegans or Drosophila melanogaster. But only about 2 to 3% of our genome codes for protein sequences, and more than 50% of it is simply repeated sequences. And these repeats might be short tandem repeats, in other words, sequences like AT, 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 or they may be large sequence repeats, so these transposons, which are like little viruses that can move around the genome. Now, about 35% of genes have alternative splicing, so that means that the exons that encode our proteins can be spliced together in different ways, generally skipping or adding a different exon in different tissues. And that 2 to 3% of coding DNA codes for somewhere around about 20,000 proteins. But given the alternative splicing and the fact that on average each gene has 3 splice variants, that means that we can encode approximately 60,000 different proteins. I should also point out that things like antibodies and T-cell receptors have a slightly different mechanism involving splicing at the DNA level rather than at the messenger RNA level, in order to generate the huge amount of diversity that's needed for the immune system. So what are we trying to achieve with bioinformatics? Well, on the left-hand side, we see the central dogma of biology, that DNA sequence encodes protein sequence, and that in turn encodes protein structure. And of course then protein structures have functions. So what we're trying to do to a large extent is understand how DNA sequence encodes protein sequence. Now, the process of translating from DNA via RNA to protein is of course relatively trivial. However, we need to know which bits encode protein. So given that only 2 to 3% of our genome encodes protein, how do we find those 2 to 3%? Similarly, given that we have this intron-exon structure for our genes, where exactly are the intron-exon boundaries? These are difficult problems, difficult things to predict. Then once we have a protein sequence, we need to be able to predict how that folds up into a protein structure. Now, that can be done based on looking at homologs, and we will come back to that in a minute. But also these days we have these methods that have proved relatively successful. This project that you may have heard about called AlphaFold 2, which uses artificial intelligence deep learning methods in order to predict protein structure, and far outperforms other methods on average. And from protein structure we have protein function, because it's the three-dimensional structure which encodes the function, so it would be good if we can also predict function from structure and indeed from a sequence at a less detailed level. And once you can understand how structure and function relate to one another, you can intervene with this, which of course involves the design of drugs. Now, I said we'd come back to homology, and this really is a key concept for everything related to bioinformatics. So, homologous proteins or genes have descended from a common ancestor. This is the definition of homology. Consequently, it is wrong to refer to percentage homology. You can't have proteins or genes that are 50% homologous or 75% homologous, just as you can't be 50% dead or 75% pregnant. Now, you can measure how long you have been dead or pregnant for, and in theory at least you could go back in time and look to see when two genes or proteins descended from a common ancestor. But of course in practice we can't do that. But you can look at the divergence of the sequences and use that as an estimate of how long ago they diverged from one another. Consequently, you should be talking about percentage sequence identity or percentage sequence similarity, which would take into account how similar particular amino acids are to one another. Now, orthologs are homologs that have resulted from a speciation event. So, for example, hemoglobin in humans compared with hemoglobin in mice. Paralogs, on the other hand, result from gene copying within a species. So, for example, hemoglobin and myoglobin, which are similar to one another in function and structure, have some sequence similarity, but have slightly different functions. They both bind oxygen, but one's for storage, one's for transport. So given that we can measure percentage sequence identity or similarity, we can use that to infer homology. So if we see two sequences that are related to one another, then we can infer that they descended from a common ancestor. So sequences of at least 100 amino acids in length with at least 35% sequence identity are always homologs, but we can see sequences with much lower sequence identity that are also homologs. Sequence similarity, where we take account of the similarity between individual amino acids, is more useful than sequence identity. Now, why is homology so important? Now, as I implied with hemoglobin and myoglobin, since homologs have derived from a common ancestor, they will have similar characteristics. So let's have a look at those two proteins. So this is one of the chains of hemoglobin, and this is the chain of myoglobin. Now, I've rendered them in different ways, one as kind of cartoons and one as a backbone trace. But that's really only so that you could see some difference between them, because when I overlay the two, you can see that the structures are almost identical. Now, these actually only have 27.7% sequence identity. But not only do they share pretty much the same structure, they also share a very similar function, as I mentioned before, in oxygen binding. So obviously myoglobin is used for storing oxygen, hemoglobin is used for transporting oxygen, but this fact allows us to transfer annotations. In other words, if we find a novel protein and we can find a homolog, then we can transfer some information from that homolog in terms of the structure being similar and in terms of the function being similar. So this idea of being able to use the structure of a homolog in order to transfer information allows us to build three-dimensional models of proteins based on homology, and we will come back to that in the next lecture. In other words, why use equipment like this? So this machine at the top here is a small lab X-ray crystallography machine, and this machine here is an NMR spectrometer. So those are typical ways of solving the structures of proteins. So why would we use these if we can just use a simple desktop computer or even a laptop? Let's move on now to the topic of data storage. Now, I'm going to make a definition of databases and databanks, and I should emphasize that this is not a definition that everybody follows, but I think it's a useful separation of different ways of storing data. By this definition then, a databank is a collection of data, normally just in simple plain text files, so things that you could look at using Word or Notepad or something like that. And the key point is that there is not a fixed associated query tool. So there's not just one way of searching the data. Now, that doesn't mean that there aren't any ways of searching the data. It means that you can choose the tool that is appropriate to search the data in the way that you wish to search it. Now, with a databank, you can download all these data and you can handle the data however you wish. On the other hand, a database is a structured collection of data hidden behind a fixed search tool. So, in other words, with a database, you don't have the option of choosing what you wish to search with. You have to use the tool that's provided. Now, this may mean that the data are hidden behind a web interface and you don't have a way of accessing the data other than using that web interface. Or it may be something like a relational database. So I'm sure all of you have used Microsoft Tools, Microsoft Office. The extra member of the Microsoft Office family is Microsoft Access, which is a relational database. And together with things like SQL Server, which is Microsoft's professional version of Access, things like Oracle, MySQL, PostgreSQL, These are all relational databases that allow you to take data, split those data up into tables, and then search. But you can only, once the data are in that database, you can only use that database in order to access the data. Now, most of the resources that we have in bioinformatics are indeed data banks, and we can split those into primary data banks and secondary data banks. So primary data banks contain raw data, raw sequence data, structure data, and so on. And there may be some detailed annotations in there as well. On the other hand, secondary data banks contain data that have been derived from the primary data banks. For example, we could look at a particular homologous protein family and look for features of that family, such as sequence profiles and so on, and these are then stored in a secondary data bank. And these are generally highly annotated with information about the family that we're looking at. Some examples then of primary data banks for DNA. I've already mentioned these. We have GenBank in the US, EMBL-ENA, which stands for European Molecular Biology Laboratory, European Nucleotide Archive, a bit of a mouthful, DDBJ, which is the DNA Databank of Japan. As I said, those contain data about DNA sequences, and they exchange data on a regular basis such that they end up containing the same information but using different formats, different deposition tools and different analysis tools. For proteins, the primary resource these days is UniProt KB. The KB stands for knowledge base, and that has two parts to it. Tremble, which is automatically translated EMBL-ENA, and Swissprot, which is more carefully annotated and hand curated. So these examples simply contain sequence data for DNA or protein. They also may have feature information, by which I mean for DNA, things like the splice sites, where the exons and introns are, and so on. We also might annotate things like signal sequences, disulfides, active sites, and so on. The DNA databanks frequently also contain translations. So these may be known translations where we know what the sequence of the protein is, or they may be predicted based on computational tools. Now, given that a lot of these protein sequences are predicted using computational tools from genomic data, unfortunately these gene prediction methods are imperfect, and therefore a protein identified from genome data without having been experimentally verified is labelled as hypothetical in these resources. A couple of other examples then of primary databanks include the PDB, which stands for Protein Data Bank, and that, as I mentioned earlier, contains structural data, generally of proteins, but also of DNA and some other things. Then there's the Enzyme Data Bank, which classifies enzymes, and so on. Secondary databanks, on the other hand, contain information derived from the primary databanks. So typically patterns that characterize a particular protein family or functional groups, and we'll come back to that later. They almost always have detailed annotation, and there are some examples at the top there: ProCite, prints and blocks. And Interpro is a resource which brings together multiple secondary databanks into a kind of unified, integrated search and analysis tool. And the point about search there is that these things can be used for finding homologs, and we will come back to that a bit later. We're going to take a break at that point, and in the second part we will look at sequence alignment, homology searching and annotation.